Hello guys, welcome to my YouTube channel. So today we are going to learn protein transport into the mitochondria. But before that we have to see a brief explanation on the structure of mitochondria. So mitochondria, as you know, it is also called as a powerhouse of the cell, and it has outer membrane. This is the outer membrane. It has an inner membrane. This is the intermembrane space, the space between the outer membrane and the inner membrane, and this is the matrix within the mitochondria. And these projections, finger-like projections, are called as the cristae. Alright. So this is the basic structure of a mitochondrion. Now if you look at specific protein structures within the mitochondrion, on the right hand side you will see that the outer membrane has one protein complex which is called as the TOM complex. Alright, this complex is involved in protein transport, and another secondary protein complex is also there which is called as the SAM complex. So these two protein complexes are present in the outer mitochondrial membrane. Now coming to the inner mitochondrial membrane, there are separate protein structures which help in protein transport. These are team 23 complex and team 22 complex. So these two team complexes are present within the inner membrane of the mitochondria. Apart from that, we have an oksbi complex, the secondary protein complex present in the inner membrane of mitochondria. Now, what are signal sequences? Signal sequences are a specific protein region with which a protein can be directed to the appropriate cellular compartment within a cell. So they are specific sequence of amino acids that direct the correct transport of protein within the appropriate cellular compartment. So if you see, suppose this is the protein to be translocated in mitochondria or peroxisomes or endoplasmic reticulum, any cellular compartment, it will have a specific signal sequence shown by this green portion at the N terminus. So such kinds of proteins which are having signal sequences at the N terminus are called as N-terminal signal sequences. On the right hand side, this is the signal sequence of cytochrome oxidase, which is a protein found in mitochondria. So the first 18 amino acids of this protein are actually the signal sequence that direct the translocation of this protein into the appropriate mitochondrial subcompartment. Now we come to the protein transport into mitochondria. So first what happened, the protein to be translocated into the mitochondria, they have a signal sequence at their N terminus. This is shown by the red portion. So this binds to the import receptors, which is specifically the TOM complex in the outer mitochondrial membrane. So on binding to the TOM complex, what happens, that this is the first step of protein transport into the mitochondria. So after binding to this TOM complex, what is the second step? The TOM complex imports the protein through this complex. The TOM complex imports the protein and it is guided inside the matrix space within the mitochondria. So this is the second step. So after so what is the second step? After passing through the TOM complex, the protein is taken up by the team 23 complex present in the inner mitochondrial membrane. So the protein first passes through the TOM complex, then through the team complex, and finally the protein arises within the matrix space of the mitochondria, which is inside the inner mitochondrial membrane. So this is the third step of protein transport into the mitochondria. So after arriving within the matrix space, this signal sequence, this N-terminal signal sequence, will be cleaved by a signal peptidase. This N-terminal signal sequence will be cleaved by a signal peptidase. It will be removed and this mature protein remains within the mitochondrial matrix space. So the proteins which are destined for the matrix space are transported first through the TOM complex in the outer mitochondrial membrane, then through the TIM complex in the inner mitochondrial membrane, and finally the signal sequence is removed through the signal peptidase. So there are other certain factors which help in mitochondrial protein transport. Number one factor is ATP hydrolysis, and number two is membrane potential. So these two are very much important for the proper translocation of protein within the mitochondria. Now, first we know that any protein which is to be destined for any compartment of the mitochondria remains associated with cytosolic HSP70. What is HSP70? It belongs to the group of chaperone molecules. Now, what are chaperone molecules? These molecules or these proteins actually help the mitochondrial precursor proteins to remain in their unfolded state before being imported within the mitochondria. So this HSP70 actually helps to the protein to remain in its unfolded state by binding to this mitochondrial precursor protein. Alright. Now, in the cytoplasm, the protein which is to be destined for import into the mitochondria remains associated with the cytosolic HSP70. So, this protein along with cytosolic HSP70 binds to the TOM complex for import in the outer mitochondrial membrane. As soon as it binds to the TOM complex for import, this HSP70 will be removed by ATP hydrolysis. So this is one step where ATP is hydrolyzed. This is the first step where it is being hydrolyzed to remove the HSP70 from the mitochondrial precursor protein. Next, it will be translocated when it arrives at the junction at this position. When it is to be translocated within the inner membrane, what happens? Here you need a proton gradient and you need a potential gradient to transport the protein further within the inner membrane. So what happens? Pumping of proton takes place from the matrix space to the intermembrane space. So as a result, what happens? The protein is imported within the inner membrane of the mitochondria from the intermembrane space through the use of TIM23 complex. Alright, so as soon as the protein is destined to be released within the inner mitochondrial membrane, immediately mitochondrial HSP70 attaches with it, the reason being the same as before, to keep it in the unfolded state before being it is utilized by the mitochondria. So this is the mitochondrial HSP70 which attaches to it, and we know that attachment or removal of chaperones require ATP hydrolysis. In the previous case, chaperone removal requires ATP hydrolysis. So here also attachment of mitochondrial HSP70, that is attachment of another chaperone molecule, requires ATP hydrolysis. So this is the second step where ATP is again hydrolyzed. And finally, it causes energy dependent conformational change in HSP70, and finally the mitochondrial precursor protein is imported in the inner mitochondrial membrane. So two things you need to remember here. One is there are two steps where ATP is hydrolyzed. This is the first step where ATP is hydrolyzed, and this is the second step where again ATP is hydrolyzed. Why? Because these two steps are removal of chaperones and attachment of chaperones respectively, and this is another step where proton is transported from the matrix space into the intermembrane space. This membrane potential is required to drive the protein import from the intermembrane space within the inner membrane of mitochondria. So if all these factors are there along with the presence of signal sequences, then only proper mitochondrial protein will be imported within mitochondria.